All right, here we go. So let's talk about what we've got going this week. There's no lab today, obviously. We talked about that. No lab today, so we'll push that lab and we'll do that next week. So if you haven't gotten your stuff done, I think most of you have. Let me know what day you want to do it. And that may be mostly people I'm talking to right now. But send me an email, let me know what day you want to do it, and I'll make sure that it is set up. So let's see, no lab today. We'll have a quiz on Friday. Stuff since the last quiz, so get the citric acid cycle. We haven't tested the pyruvate dehydrogenase complex yet, right? I think stuff from today is reasoning. I think that it's not a big memorization test, so I think that is also going to be fair game. Let's see, I need to hand back your quizzes, maybe remind me and stop in the last 5 minutes for that. Questions on what we're doing? Otherwise? All right. Okay, so let's go ahead. What I want to do is I want us to start thinking about energy here, okay, in terms of the energy produced by these pathways. I've given you numbers so far. You guys hopefully have your glycolysis figures out there as well, but I want to talk about something called phosphate. It's called PO ratio. And what it means is it means phosphate to oxygen ratio. And essentially, it has to do with how many ATP you can make from a given molecule in the electron transport chain. So it's how many ATP can be made from a given electron donor in the electron transport chain. Yes.
This is a random question. Are you having a quiz on Friday? Yeah.
Yes. Okay. So essentially, what that means is we'll go over the electron transport chain later, but not all of these molecules are going to give you the same amount. And also, you can get depending on what's going on, you can get more or less of these. So let's say somebody has hypothyroidism. Some of you may be familiar with that. Symptoms of that are you cold, you're tired, and things like that. And your electron transport, what's going on in hypothyroidism is your thyroid is not making as much T4, which signals how active your electron transport chain is. The more active your electron transport chain is, the leakier it is. Okay? So actually, people who have hypothyroidism, their electron transport chains are more efficient. All right, so if you have hypothyroidism, you make 3 ATP per NADH instead of making 2.5 ATP per NADH, okay, because things are less leaky. But what happens out of that is consequences if you're not running as much stuff through, if that makes sense. Okay, so you could think about the electron transport chain getting sloppier the faster it runs. But if we think about the electron transport chain, we'll make it less complex, or we'll make it more complex. But if this is the electron transport chain, and we think about, we have our NADH, which can enter over on this side. So remember, it's a PO ratio. It's per oxygen. So it's essentially per oxygen we put over here, how many ATP are we going to get out? So we get NAD plus here. That enters right here at the beginning of the electron transport chain, so you can capture more energy going down. If we think about ubiquinol, remember ubiquinol, we have an FADH2 that gets made and it transfers its electrons to ubiquinol. Ubiquinol enters later. So what we do is we miss we miss capturing energy here and using it to pump protons across the membrane. Okay, right at the beginning of there. So you get less from a ubiquinol. So when we talk about PO ratio, under normal conditions for usually NADH is equal to 2.5 ATP. I'll kind of always give you your PO ratios. You don't have to memorize these, but they're kind of interesting for keeping in mind. So, and ubiquinol is worth 1.5 ATP. All right, so it's worth less in terms of the amount of ATP in this case, right? Remember this came from an FADH2? And you remember in the pyruvate dehydrogenase complex that we actually, that FADH2 actually gives us, we can make an NADH from it. So that FADH2 is worth less in the citric acid cycle than it is in the pyruvate dehydrogenase complex. Remember we talked about FADH2, the amount of energy it contains is based on its environment, sort of. Anyway, so it can the QH2 is always going to be worth the same amount because it's a free floating molecule, though. Questions? All right, so now what we have is, now that we know this PO ratio thing, we have the ability to go through our citric acid cycle and our glycolysis, and go through glycolysis and calculate the amount of ATP we get from something. Okay, and if I was to change things up, you would be able to also calculate how many ATP we get from something. So I give you some other thing you start with. That make sense? I could also give you an alternate universe problem. All right, so we've got those numbers from up above. I'm going to put the citric acid cycle up here as big as possible. Haven't given you one of these diagrams yet. And I'm going to hope that you have your glycolysis diagram, or you have memorized it so much that you don't really need this much. But you can ask me questions about it, okay? I'm going to leave it on because I can make the other thing big. But let's go ahead and let's look at this. How many NADH2 do we get off, or I'm sorry, NADH do we get out of the citric acid cycle? One round of it. 3, right? You guys see that? And then how many ubiquinols do we get out? 1. You already got that? And then what do we see at the bottom that's in the little yellows? Can people see the little yellow star? A GTP per cycle. So what I'd like you to do is, if you were given a glucose molecule, right, how many ATP do you expect to get out of the complete oxidation of glucose? I want you to calculate it. You know it's 32 because I've said it a whole bunch of times, right? But in this case, how many would you get out? And what I want you to do is I want you to break this up, especially, when I ask you this on a quiz, I want you to break it up into the parts. So we're going to get ATP from glycolysis. All right. And you could take, in terms of thinking about the ATP you get from glycolysis, if we make an NADH in glycolysis, or maybe 2, those go on the electron transport chain, I would count those. That make sense? All right. So what happens between glycolysis and the electron transport chain? I'm sorry, and the citric acid cycle that gives us some maybe ATP. Yep. So ATP from the pyruvate dehydrogenase complex, and then we've got ATP from citric acid cycle. I want you to try that. Okay.
Is this good for glycolysis? Is it net ATP?
Net ATP. So you're going to lose some and gain some, right? Okay. But in terms of thinking about that, like I said before, I hope you have this to pull out because that's going to be hard to see. This we just talked about what's there, and if you've forgotten, just ask me. I'm going to be walking around bored anyway. Okay, so ask questions as you have them. But this, right? You get in glycolysis, you're also getting, you get the ATP that come out straight, but you also get the NADH. And what we're saying is our PO ratio our PO ratios are going to be NADH equals 2.5 ATP. We're going to run with that. And QH2 is equal to 1.5 ATP. And that's all we need PO ratios for.
GTP count as
So any of the NTPs are equivalent to 1 ATP, and they're designed to just keep them at various concentrations. Actually, I don't know how they take no energy in the reversible reaction, so I actually don't know how the concentrations are regulated. But, yeah, but they are. They're regulated at the concentrations that are needed.
Are we also taking into account, like, we're breaking up one molecule, so it's going to be technically twice?
Yes, so are you checking, absolutely. So that's a great comment. So I just had a comment about, so you take glucose and you make 2 pyruvate, right? And those 2 pyruvate, so how many times per glucose do you run through the pyruvate dehydrogenase complex? Twice. And then how many times do you run through the citric acid cycle? Twice. In terms of checking your answers, you should get 32 at the end. If you don't, I'm happy to look at it.
What do we, when you say pyruvate dehydrogenase, is that on the board?
It's not on the board, but if you remember having your notes, what you get your energy out of.
Yeah, it's
This pyruvate comes from pyruvate to acetylcholine. You make an NADH.
Is it because we're not counting by?
Are you off by a certain number?
I got 20.
You got 29. Okay, so let's take a look at it. So you've got, so 29 means they're up by 3, which is interesting. Kind of a GTP, right?
It's also possible I
You've
did
got
not.
2 of those. You've got 6 of those. I agree with that. You've got 2 of those. You've got 2 of those. You net 2 ATP out of glycolysis. Yes. So then 7 ATP for glycolysis. That's from the 2.5 and 2.5.
Because I don't know how to do them.
No, it's not.
No, so, no, yeah, because 2.5,
Yeah, 2.5 and 2.5.
So then,
that gives me 5 plus
Do I
2.
have much equal 29?
no,
Okay.
there's
Okay.
7.
20.
Because we have to get NADH.
Then I knowledge.
Did I count what? Yes.
And then you choose,
And you the QH2 is 1.5.
Mhm.
and then that's 10.
I'm not looking at your notes, I'm not sure. See
So I
where
just did math wrong.
there's possibly one of them. They should be hurt.
Because I wrote it down.
Let's go ahead and work. Let's go and work through this one. And the way I like to do this, I'm going to move away to make some room here. I'm going to move away from the pathways. But so let's say we've got glycolysis, right? So glycolysis, when we think about it, we get out of glycolysis, we're going to get. So you can think about we net 2 ATP, right? So we get 2 ATP directly. And we break that, we get 2 NADH, right? Because it splits up into 2 pyruvate. And then if we look at this right here, we get two glyceraldehyde 3-phosphates, and we take them through, and that's in this step going to give us, for each one, an NADH. So I've got 2 NADH, and I'm going to I would multiply that times 2.5 because that's our PO ratio, so that's going to give me 5, and so glycolysis is going to give me 7 ATP. All right, now let's go ahead and do the citric acid cycle. No, let's not do this. Let's do the pyruvate dehydrogenase complex, because that's next. Okay, that's between glycolysis and the citric acid cycle. And we have how many pyruvate per glucose goes into it? 2. So we're going to end up with 2 NADH times 2.5 gives me 5. So I'm going to get 5 ATP there. All right. Then we have the citric acid cycle. We're going to make 3 NADH times 2.5 or 7.5 ATP. We're going to have 1 ubiquinol times 1.5 or 1.5 ATP. And then we're going to have 1 GTP, which is equal to 1 ATP. And then we're going to have to multiply that all by 2 because there were 2 pyruvate going through, or 2 acetyl-CoA are going through that. So this is going to give me what? That gives me 9, 10. Is that 10? 10 times 2 for pyruvate times 2 acetyl-CoA. Or we could just say times 2 cycles. All right, so that's going to give us 20. So then I look at that and I've got 20 ATP. Then I add them all up. 20 ATP, 25 ATP plus 7 is 32 ATP. Okay, questions? You should also be comfortable with, if I was to change these numbers, you should, I say, okay, we've got this condition where we're getting or an alternate universe where our PO ratios are these or this or that or the other thing. Okay, can change that or come in at a different point. Let's take an example of what a different point might be. Okay, I don't have it here, but let me grab our gluconeogenesis. bless you. All right, it's going to be another one up. Here we go. Let's take a look at this. All right, so here's gluconeogenesis, right? Where it's got glycolysis and gluconeogenesis. I'm concerned with the bottom down here. There may be an organism that does this, but I'm just going to say it could be alternate universe. An organism consumes oxaloacetate. Check everything. I'm just having a hard time spelling today. Harder time. But an organism consumes oxaloacetate. How many ATP are made if the oxaloacetate is completely oxidized? All right. Assume PO ratios of 2.5 and 1.5. All right, so we have the same things as before. Hopefully in your notes you have how many NADH and what we have for the citric acid cycle, right? So how will you consume this knowing this? These are your pathways that are available. This and regular glycolysis and citric acid cycle. Anybody have an idea?
I'm confused by like what you mean.
Okay. So this organism has these pathways available, are regular pathways available. It's going to consume oxaloacetate and it's going to completely oxidize it. How do we get from here to carbon dioxide and water? And then we take all our we take all our cofactors and run them through that we make and run them through the electron transport chain.
Well, wouldn't you just do like, I don't know if you can start with pyruvate kinase, but like just do the opposite of that and then go from the pyruvate as normal.
Start with oxaloacetate. Oxaloacetate goes to what? Phosphoenolpyruvate, right? So we're gonna go here, right? And then we'll go to here, and then it's our then it's the rest of the process as usual. All right, gotta calculate that. So it's per oxaloacetate. How many ATP do we get?
It's 16.
Is it 16? Okay.
I think so.
Wait, what? So what? I mean, isn't it just there's ATP? You're losing ATP. So is it more to ATP put on ATP?
What was that?
Like,
oh, here.
Yeah, just
here, right.
Look at this GTP.
Great question. So here you're going to use a GTP, right, which is equivalent to an ATP, and then you're going to get an ATP back.
But then pyruvate going to oxaloacetate.
Then the pyruvate Where does the pyruvate go after that?
So you just.
Then the pyruvate is going to go through the electron transport
The
chain.
pyruvate. So
Right.
it's just everything minus glycolysis.
That's
Not from that, but then you can gain.
Not from glycolysis, but you take the pyruvate and then you take that down through the electron transport chain.
So you get 25.
Yeah, I'm going with 15 without doing the calculations.
Where's the extra 10?
You still get what you get in.
What was that?
You get 5 from pH.
Oh, you have to take the subtract the NAD.
Do you use 2 GTP here? You don't. You use 1. This per oxaloacetate is my question.
But wait, shouldn't you still get the 20 from these?
From what?
From the NADH, the 6 NADH.
Yeah, that's one. But you don't get
But
20.
you don't have
just
2,
get 10.
you have it.
You just get 1.
So you've got
Yes.
1 here,
Yes.
right?
But then you would also.
So
have
1
to-
here,
When you also then have to
so
split
1 here.
that, you only get one of those, so then it would be
For
12.
TCA.
From the NADH from the pyruvate hydrogenase complex, so you would get 12
Still
to 5.
Right.
You still
Oh,
get
no. Phosphoenolpyruvate is 3.
It's one NADH per
Pyruvate is
and so
3.
one pyruvate.
So we lose this carbon dioxide here,
12.5.
right?
Yeah, it's 12.5.
Is it 12.5?
Yeah, because you only get one NADH from the pyruvate dehydrogenase complex because you only have one pyruvate.
12.5.
Yep. Oh, well,
Collaboration, we got there eventually.
okay. Well, we're going to go through, let's go ahead and just walk it through the whole process because there's some more. So what we've got here is we've got an oxaloacetate, right? So let's just talk about, I'm going to call it glycolysis gluconeogenesis. All right, so that is right here, and it's what we just did. I did with the red arrows here. We've got 1 oxaloacetate. It makes 1 phosphoenolpyruvate. It takes 1 GTP to do that. So we're going to do minus a GTP, right? And then we're going to, coming back down from phosphoenolpyruvate, we make 1 ATP plus 1 ATP. So overall, it's going to be 0 ATP. Is everybody comfortable with that? All right, so now we have the pyruvate dehydrogenase complex. We need to make this into an acetyl-CoA so we can put it into the citric acid cycle. So you got pyruvate, oh my gosh. Sorry, it's like the cat's sitting on my keyboard, but the cat isn't here. All right, so pyruvate dehydrogenase complex. We make 1 NADH, right, when we go from pyruvate to acetyl-CoA. So we have 1 NADH, and that's going to be equal to 2.5 ATP. And so we get 2.5 ATP. And then we're going to do 1 cycle of the citric acid cycle, which is, you're right, it is 12.5. So one cycle of the citric acid cycle, which is 3 NADH, right? It's 1 ubiquinol, and it's 1 GTP. Yeah, whoops, not equals. So this times 2.5 is going to be 7.5. This times 1.5 is going to be 1.5. This is going to be 1 ATP over here to give us a total of 10. And then 2.5 ATP plus 10 is going to be, we're going to get 12.5 ATP total. Questions?
And that's because, like, that organism consumed oxaloacetate, so it's only going through the cycle once.
It's only going through the cycle once because we're talking about 1 oxaloacetate. Oxaloacetate isn't part of glycolysis, right? We're already part of that split. We don't have 6 carbons in oxaloacetate, if you think about that. But the deal is you want to walk through the cycle there. We want to walk through and plan out what happens here, and then what's going to happen to my citric acid cycle based on whatever the problem is. If I changed your PO ratios, you'd have to change the numbers in the citric acid cycle, right? However, and oh, don't forget the pyruvate dehydrogenase complex. Yes.
So why do you not include anything from glycolysis?
Why do I not include
Yeah,
for this right here?
yeah,
Because I could just make this work this way.
okay.
Right.
We just start with the opposite.
I mean, I could push it all the way up, right, and pull it all the way back down, which would actually lower my amount of ATP. It wouldn't. I might say, if a problem, maybe I'd be careful with a problem and say it in the most efficient manner using the pathways you know. But there's no reason for it to actually. This is a reversible reaction. So you make this, just come right back down, right? Unless this is turned off. Questions? So what I might ask you, your organism is consuming glyceraldehyde 3-phosphate. How many ATP are made? Okay, and so in which case, per glyceraldehyde 3-phosphate, that needs to start here and run your way down for one of them. So I want you to be comfortable with those skills, adding up ATP from these things. Okay? Now let's go ahead and I want to talk a little bit more about the citric acid cycle and some other uses of it. The citric acid cycle is actually a great source of intermediates. Actually, I should say both. Both glycolysis and the citric acid cycle are good sources of intermediates. Okay, and so this will happen in normal cellular metabolism, but in cancer, you'll actually see these cycles being upregulated. Okay, so what do we know about cancerous cells? Yeah, so cancer cells are rapidly growing and dividing cells. So they need, what do they need? Energy. Okay, and energy they're going to get from this, right? They also need what? Building blocks, raw materials, right? They also need materials to grow. Okay, and there's one other thing you can think about that they need if they're rapidly growing. Space, they go find the space, right? Eventually they acquire mutations that allow them to chop through connective tissue so they can get out. They need a blood supply, they need oxygen, things like that, right? But
I mean, like they're avoiding apoptosis.
they're avoiding apoptosis, they need to turn off apoptosis. These are all important mutations that you see in cancer. But a thing that they need that's more metabolic that I'm thinking about is they need antioxidants, actually. They've got this ramped up metabolism, and actually what happens in cancer cells, if they don't ramp up antioxidant systems and repair systems, they end up, because remember, regular metabolism makes these hydroxide radicals, that they end up with things getting torn up. And actually their nucleotides end up being damaged and they'll have problems. So that's a place you can get. They need antioxidants as well. Okay, and so this is something we can get from glycolysis in the citric acid cycle. Okay, so materials, great source. Energy is something you can get from the citric acid cycle. Antioxidants and glycolysis. Antioxidants, what do you think?
We get them from not turning on, like not getting NADPH from
Well,
glucose phosphate.
so you want to get NADPH,
Oh, yeah,
right?
yeah.
Yeah, it's antioxidants. So this is the pyruvate phosphate, thank you, pentose phosphate.
Like it not working.
No, working.
I also thought this-
Wait, but doesn't that like shut off?
It sets off oxidative damage.
Is that making sense? Sounds like there's
So
some,
antioxidant, that's like when it's not okay.
right. The antioxidants is anti-free radicals.
Okay, I'm sorry, can you explain again why I don't really understand the connection between that and how that helps cancer?
How having so in cancer you have a lot more metabolism. Just general metabolism will make oxidants. Okay, make superoxide radicals that turn into these hydroxide radicals that will tear stuff up. Okay, if you have a lot of them, if you have a lot of them, you end up with with jaundice. But less of them, what happens is they're being made in the cell. They're next to the DNA. They're next to the nucleotides. Actually, where I've seen in the primary literature where I've seen effects that are useful is there are enzymes that repair damaged nucleotides, either repair them or remove them, so that they don't get incorporated into DNA. But if you take if you turn those off, you can selectively inhibit the growth of cancer cells over normal cells, or selectively kill them, because the cancer cells go ahead and then they're using the nucleotides that are damaged. Does that make sense?
Yeah.
So that's just an enzyme that recognizes and repairs. That's not against rid of the antioxidants. But you need to make more antioxidants as a cancer cell if you're going to be successful. One thing to remember about cancer cells as well is if you have like a tumor, so you've got a tumor here, the tumor started from some cell in there. The cells out here aren't necessarily the same as a cell in there. I mean, they're the same type. They came from the cell, but they're acquiring mutations as they go along. So if I'm a cell out here, these cells are competing with each other too. They're not cooperating to make the tumor, right? They might make signals to make blood vessels come in, but they're kind of doing that on their own. They're not doing it to help out the other cells. So cancer cells are all competing with each other. And so if I'm this cell right here and I suddenly get a mutation that causes my antioxidant system to ramp up, now I have an advantage over all my neighbors, and I can grow faster. And it's the fastest growing cells in that tumor that are going to dominate over time. So in that tumor, there's a lot of natural selection going on. Okay, so what I'd like to do is talk about how these sorts of things can be used. I guess the first figure I have here is that citric acid cycle intermediates can be made from common molecules in the cell. I'm going to say from amino acids and intermediates from other pathways. All right, and so this shows some of the common ways that happens. In general, this is a really important one, the pyruvate to oxaloacetate pathway. That's an important way if you want to make more citric acid intermediates. If I make more oxaloacetate, well, there's no way to make extra malate. There's no pathway here shown for malate. If I make lots of oxaloacetate, does that help out with malate?
Yes.
Yes. Why?
Because it's reversible.
because you've got a double arrow there, right?
Is that why they
Right.
have reversible steps in the citric acid cycle?
I don't know that. So the question is that why they have reversible steps in the citric acid cycle? Generally enzymes are catalyze reversible steps. You get lots of enzymes that catalyze reversible steps. So um, these, we have to have these. If we're going to have the citric acid cycle go in this clockwise direction, we have to have these 3 irreversible steps, right? Or at least 1 irreversible step. I don't know about irreversible steps from a natural selection point of view. I'm not sure. Okay, but if you make more oxaloacetate, now you can run the citric acid cycle more. You can run it faster. And non-cancer cells do this too. They're going to ramp up and down depending on levels of activity. Okay. So amino acids. Yeah, Sophia.
Okay, yeah, so what if the oxaloacetate not just go to the pyruvate? Like it's in both places?
It's in both places,
So
yeah.
Is it like by the location that it does what it needs to do? Like, do you know what I mean? Like how does
Right. So in terms of oxaloacetate, well, it's an intermediate in the citric acid cycle, right? So you just kind of run through it pretty quickly. Or not citric acid, I'm sorry. Gluconeogenesis. So in terms of gluconeogenesis, you're going to use, you have an enzyme right there. It'll take pyruvate, it'll turn it into oxaloacetate, right? So you can generate it from that. How it's regulated in terms of, there must be a shuttle because the citric acid cycle happens in the mitochondria and glycolysis happens in the cytosol. So I guess it's going to be controlled by what compartment it's in.
Yeah.
But while I would expect there to be, a reasonably standing amount of oxaloacetate around in the citric acid cycle, right, in the mitochondria, I would not expect it to be for gluconeogenesis, because I would expect that to just kind of rip through and go. Does that make sense? I expect those concentrations to be held fairly low in the cytosol. I do want to show you something with oxaloacetate here, though. So, biochemistry is a giant memorization fest, right? But I try to reduce it as much as possible. If we think about the amino acid, sorry, I'm doing my amino acid wrong here. If we think about aspartate, interesting. I don't know whether I'm going to be able to draw aspartate here. That's a good idea. Yeah, I don't know what's happening here. I'm going to close, see if I can get an arrow and I can close my OneNote. Close, please close. There we go.